start. Lu, anytime you want. Whenever you want, it's fine. I think it's already time, right?
You ready? I'm ready. Can start?
Yeah, please. It's your call.
Us. So, my dear friends, hello, Shallow Lakers. Thank you for coming to the special session, Ecology and Energy Flow and Tropical Temporary Ecosystems. The special session coordinator, it's me and Dr. Clarice Casanova. Before to start, I'd like to check our microphones and camera. Is everything okay? And so we hope you are enjoying our special session. Firstly, some advices. Please leave us your questions and comments in the Q&A, or questions and answers. Don't forget to put your name, institution, country, among others details. Speakers or presenters, I'd like to kindly ask you to keep your microphone and camera off. Session coordinators and presenters may leave these devices opened. Finally, our session will be recorded. It's okay? Recording? Yeah, recording. So, we are a few people, but it's no problem. We have a great discussion to do. And well, enjoy our session and discussions. And I'd like to start. I'm Luciana Gomes Barbosa. I'm professor at Federal University of Paraíba. And I'm a founder and currently a chair of International Network on Limnology of Drylands. Our main objectives is understand and identity the distribution of aquatic ecosystems in these regions around the world, the main vulnerabilities, among others, and factors that affect our ecosystems, aquatic ecosystems. So I'd like to introduce my dear friend, Dr. Clarice Casanova. Clarice, please.
Hi, everyone. Again. My name is Clarice Casanova, as Luciana already told you, and I am an associate researcher, maybe a postdoc, right, in the Federal University of Rio de Janeiro. I'm glad to be here and in this special session, which is covering like a big field in temporary ecosystems functioning and regulation. And I think it's going to be very prolific, our talk here, even though we're few. It's going to be nice, and we can talk a lot about our things and what to expect of them. Because what I presented in the YouTube video that was sent, it's part of my PhD. I'm still processing some material from my PhD from 2-3 years ago. And one of them, this part is talking about humic substances in very shallow and temporary ecosystems in the Restinga of northern Rio de Janeiro state. So it is still a black box. Actually, it is a black box because there's really a lot of humic substances. So we have a lot of very dark waters in this place. So it's still a very black box of what's happening within this ecosystem and how communities are being regulated since the values we encounter of dissolved organic carbon in this place is off the roof, it's very high. So the work I presented was trying to figure out how energy flow is working in this ecosystem, and especially trying to connect with fatty acids quality for zooplankton. So actually we got to answer some questions related to ceston quality, actually, because ceston quality changes a lot during the filling state and the section state in these temporary ecosystems, and that was not different for this particular ecosystem that we studied. And we can see that the relation between fatty acids, the good fatty acids, the very yummy fatty acids for zooplankton, is related to the presence of cryptomonas, which is very and highly nutritious for some zooplankton organisms. And this is very interesting, though we have still to do some things to still process those samples and maybe go a little bit deeper on how alloctonous or autochthonous this source of energy is. And so we probably in the future, we're going to have to conduct some stable isotopes analysis to see whether it's actually mostly the part of zooplankton are mostly ingesting is autochthonous and highly nutritious like cryptomonas, or if it's actually like plant debris or detritus or bacteria from alloctonous sources, since it's a major contribution in this particular ecosystem. So that's pretty much it. And we also found that because this study I presented was actually part of a bigger picture, which is the study of different ecosystems, also shallow ecosystems, but not temporary, in the northern Restinga area. And what we see is the dependence of this highly nutritious consumption of autochthonous origin in the temporary systems, and not so much in the non-temporary systems, but those shallow lakes as well. So it is in a bigger picture, we can see that it's extremely important to try and understand how different this temporary ecosystems work, how differently they work. So yeah, there's a lot to talk about still, but let's move on, or else I can monopolize all the talk.
Oh, no monopolize. Very great talk. Yeah, I like it.
Right.
Because
Yeah.
this session is the first step to discuss about it, because I see several gaps in temporary ecosystems. We need to study more and to introduce deep questions, because the functioning and energy flow and the response of aquatic communities is very high, and we have more databases maybe in the Europe or Mediterranean region, but around the world, the data from temporary ecosystems is very small. So we need to understand better, and it's very important, this meeting in this Congress. And we can start with the Luisa, Costa, and Rafael. And after that, we can speak about our works. What do you think?
Yeah, sure, good. But please turn on your camera so we can see you.
Yes, we need.
Don't be shy. Don't be shy.
Don't be shy. Rafael, then Luisa, please open. Oh, good, good.
My camera is working?
Not yet, Rafael. Just a second.
Yeah, it'd be very interesting if you can summarize our study in 5 minutes approximately. Please, Luisa, and after that, Rafael.
Yep.
Okay, I can start. Are you listening? It's okay?
Yep.
So my work was about characterization of natural and commercial humic substances, because I'm related to Clarice. So I work with a little bit of the line of research from her. Well, in this work, I use the methodological approach to see if are there differences between natural and commercial humic substance content, and we use chemical characterization, we use photometric, I don't know. We use absorbance, it's better, absorbance readings. We use emission and station readings. We also use SUVA index to see the difference between these two sources. I think I can use this word, two sources of humic substances, and we saw that when we look for each characterization, for absorbance or for emission or station, we had a different answer. But if we see the bigger picture, if we integrate all these answers, we had different and an answer that we can extrapolate. So when we saw absorbance readings, we saw that natural and commercial humic substance were pretty similar, and if we stop with this characterization, we could think that they are similar and that we could use commercial humic substance to mimetize the natural condition. But when we use we go to the next step, and we saw SUVA index to see the aromatic content, or we use emission and station readings, we saw that they are completely different. The reactivity compartment was very, very different. So in the end of the work, we conclude that we had to make a little integrate approach to characterize these two contents. And I think I didn't tell, but the natural environment that we used was a temporary wetland from Restinga to from Rio de Janeiro, and we saw that there are a lot of difference. And after that, I kind of started to looking for this difference and look for the difference in chemical functioning of dissolved organic carbon and wetlands. And I saw there was nothing, because nobody was studying this environment, nobody was looking for the characterization of chemical structure, chemical functioning, chemical changes. So after this work was a simple work, we saw that there are differences. We saw that we need to integrate the answer, integrate the approach, but there is only a few things, a few works that looking for it. So it was a beginning for searching and for looking about the chemical functioning of this environment. I think it was that.
Cool. Well, Luciana, let me just ask Luisa, please, tell us, are you're an undergrad? Did you mention that? I'm sorry. Just so background.
No.
Quick background check.
It was I was sketching in a hurry, so I didn't tell it. But I'm Luisa. I think I didn't say my name. I'm Luisa. I'm an undergraduate student from Federal University of Rio de Janeiro, and I work with wetlands at Limnology Lab from Federal University of Rio de Janeiro. And I'm oriented by Rinaldo Bozelli and co-oriented by Dr. Clarice Casanova here in this room.
Thank you, Lu.
Rafael,
Hello. Are you listening and watching? It's okay? Camera and audio? Nice.
There is a bit of interference, Rafa. There's a little bit of interference, maybe a ventilator or something you're using. Maybe the plug in the...
It's better now?
No. Now I can
No?
hear me. But it's okay. I think it's fine. It's not that big of a deal, actually.
Sorry. Well, I'm doing PhD at Federal University of São Carlos, and I was about to study my environment at temporary environments at semi-arid in Brazil, when suddenly the COVID outbreak stopped everything and everybody in the world. So I can only show a piece of this study in shallow lakes. It's a preliminary characterization of some ponds during 3 days period, because we had some issues and difficulties in the field, so we cannot stay more days. And we sample the ponds to analyze preliminary characterization, physical and chemical, and chlorophyll. And I addressed a question about, which is how we are underestimating phytoplankton biomass using chlorophyll in tropical systems, because we used to use bases of glacial and in temperate regions, like the formulas of Eric Jeppesen, for example, which is phytoplankton biomass based on chlorophyll multiplied by a ratio. And this ratio is could be different for tropical systems. And I kind of show this in this study. So, and I also sample phytoplankton, but I still don't have the results from this because we stopped to work in laboratory and stuff. And so I will further analyze phytoplankton biomass based on properly phytoplankton measurements to compare with chlorophyll results too. And I think it's basically this. And I'm also interested in invasive species and how they can adapt in these extreme environments. So this is a topic to address in my PhD too. And also mixotrophic individuals, such as Serratium fucoides, which is spreading to Brazil. And it's basically that.
Thank you, Rafa. I wanted to ask you something. Did you, and in your PhD, you're also working with shallow temporary waters or no?
Yes.
Yeah. Is this part of this with the semi-arid?
Yeah, because I'm studying the influence of climate change and invasive species on ecosystem functioning. So first of all, I want to understand how this system works and function to then address the impact variables like species, invasive species, and like droughts or...
Thank you. Very nice.
So, in your video or in your abstract, you say we'll need to revisit the productivity models in tropical systems, or things like that. What do you think regarding to models adaptations? Because in semi-arid regions in Brazil, for example, the rainfall is very irregular in time and space. So what do you think about the adaptations? Because it's very interesting to us to discuss. The most part of the models was produced by the temperate regions, temperate ecosystems.
Yes. Based on based on what I was reading about Reynolds in his book Phytoplankton, and he established a table of how each phytoplankton species contribute in terms of biomass, and it's not correlated to the size or correlated to a type of species. It's variable between species, and it's established for marine phytoplankton, not freshwater phytoplankton. So there's a lot of things to balance in these models, I think. And first of all is to not readily use formulas from glacial. Like I'm not here to say that Eric Yepesen is wrong, because he's working in glacial areas, but we need to think about it. And I think chlorophyll is the most important variable to address questions about climate change, about also dissolved organic carbon, Clarice, but chlorophyll is very important.
Yeah, actually, it's the first most important. The second is humidification. So we're on the same page. It's fine.
Okay. And yes, I don't know if I answered the question, but we still have a lot of work to do on this.
About the stratification, because I'm very curious about your observation in these ecosystems. You see long times without stratification or short-term thermal stratification. How long is the stratification in your ecosystem?
I think I captured a picture of a framework, because these days I was sampling, I noticed and asking people around too that the climate was very different. There was a lot of wind, a lot of the temperature was the daily variation was very high, was higher than the normal periods, and a lot of winds, and also the water was not that turbid. It's not clear, but was with sac disc showed depth considerable. So I think I captured outlier maybe. So I want to do the same thing again in other periods to understand this variation. But it's interesting to talk about this stratification, because there is a temperature variation, high temperature variation between the strata.
Yeah.
It's interesting. It's crazy. I never saw something like that.
It's crazy. Probably it's a temporary.
A short column. Yes, a short column with higher variation. It's sorry.
The air temperature is very high in these regions. Probably the stratification is short. No, yes. Daily stratification, and yeah, but very interesting. So we'll need to present our works, Clarice. And we have a question for Luisa also.
Yeah, I think so. But I have to present my work?
Yes, first.
Yeah, okay.
Present your
So-
work, and after that we can speak more about your work and
Okay.
about the Luisa.
Yeah, how it, yeah, okay, how it speaks. So yeah, as I mentioned, we did some samplings on 2 different time periods in 2015, 2016, with the objective to see whether what was shaping food quality in temporary ecosystems in northern Rio de Janeiro state, in the Rixinha area, which is a very unique area itself, with a lot of different kind of alloctonous resources for inside the water column, and how this is metabolized and how it is how it shapes aquatic communities functioning and ecosystem functioning was still a very big question mark for us. So we tried to sample this in this particular environment, which reaches up to 180 milligrams carbon per liter of carbon, which it's a lot, considering when we have in current literature people publishing papers in temporary and permanent shallow lakes in temperate regions, where they mention highly humic environments that reach up to 40 milligram carbon per liter. So it's about 3 times higher, depending on the time we sample it. So when I say it's extremely high, it is extremely high. And we can see it through the pictures that I posted on the YouTube video as well. So that was the idea. The idea was to see if this amount of dissolved organic matter was shaping and was influencing food quality for zooplankton in this shallow temporary ecosystem. And it's quite interesting because what we found was that we had a lot of high quality cestin, even though we predict that food quality, like phytoplankton, would be very depleted in ecosystems with such high portion of shading, because you have a lot of humic substances which are colored. So we predict that phytoplankton is going to be depleted because there's not enough light in the water column. But that is actually not what happens in this ecosystem. And by the time we went there in December 2015, December 2015, we saw that actually when you pass your fingers through the water in the upper layer, like this reaches about 1.3 meters of depth when it's entirely full. But in the first centimeters of water, then you can actually get all the algae out of your hand. So we had a lot of filamentous algae and a lot of different things attached to the water surface. Actually, some zooplankton as well attached to these algae, actually eating them, because as we imagine the water column and the deeper layers, it has a lot of problems with algae concentration, and chlorophyll is very low in these deeper areas. This is in another work we did, but but it's a bigger picture, is that cestin quality was important, and autochthonous resources was the most important thing for zooplankton in this highly humic environment, which is not what I expected at all, because we have a thing on ecology, we have a debate on ecology whether DOC may be nutritious enough to fuel secondary production in lakes. And some people say no. So Michael Brett group that works with fatty acids and food quality for zooplankton, and my collaborator and used to be co-advisor Dirte also agrees with that, because dissolved organic carbon, humic substances do not have what it takes to make primary consumers grow and reproduce optimally. So there's a problem with DOC, and a lot of DOC would cause depletion on this secondary productivity in aquatic environments. On the other hand, we have some people like Stuart Jones and others that debate on whether actually there's a lot of DOC, so zooplankton is eating this DOC, regardless whether if it's better or worse for them, just because there's a lot. And it doesn't causes that much depletion, and they can actually grow a little bit, but not optimally. So it's not great. They're still doing it. But what we see is actually the amount of eicosanoic pantonoic acid, which is a marker for primary producers, so phytoplankton mostly and cryptomonas mostly, found in zooplankton it's probably where they are getting their food from. So it's highly nutritious phytoplankton in this very rich DOC environment. So this is what we found in our study, that we actually need nutritious phytoplankton, nutritious cessin for zooplankton growth and reproduction. And actually, when we get to sampling zooplankton there in those lakes and sorting them, because to do the fatty acid analysis, we need to sort them individually. So there's a lot of zooplankton. So they're growing, so they're reproducing. And it's quite interesting to see that, because when we see the algae growing on the surface layers, the ones we can actually get, the filamentous one, they're not probably very edible for zooplankton. They're probably not eating those. But what they are eating and what we found, it's mostly related to the cryptomonas and cryptophytes that are flagellates that are using the light resources, probably using the light resources from the upper layers as well. So this was also quite interesting. And it shows like a very interesting way this temporary system is working. Though we only sample 2 phases, and this is that's why probably we need longer to investigate how seasonality will shape this community and how it will shape resources obtention for zooplankton, because we have the filling state and the lower water state, that this would probably change the way zooplankton is getting these resources. So it has a little bit more to be investigated in these temporary ecosystems. And there's a lot of nice questions regarding that. And maybe Luisa can go next, because this is actually something we're trying to investigate, because when she tries and see whether there's difference in humic substances reactivity, we're actually trying to see whether this can cause any kind of problems directly to zooplankton or primary producers in the long term. So yeah, that's pretty much it. And actually, this other guy just entered the room, also works with that, but he's in the resting stages of the zooplankton, so it's quite nice. Yeah, go ahead, Lou.
Very interesting. And you saw the beginning about the black box.
Yeah.
Oh, yeah. That big black box, energy fluxes and nutrients fluxes in temporary ecosystems. We'll need to include that in our studies, mainly in broad scales and long-term studies, because the fluctuation over time, reduction of the rainfall, increased evaporation in drylands regions, for instance. So this comparison between more humid temporary aquatic ecosystems and more dry temporary aquatic ecosystems may be very interesting also. Well, I can speak a little bit about my work, and this work was developed in the rock pools very well distributed around the world. It's shallow, small, and very good to test ecological theories. And I like to work in rock pools, and it's very interesting specifically to test meta-communities studies. And so we work in a small city in Paraíba State, semi-arid region, and a set of urban rock pools and rural rock pools. Rural rock pools more protected. And we try to investigate about the impact of rainfall on the phytoplankton communities. So among the main results, stochastic rainfall in this region, rainfall is high and great volume in small periods. And stochastic rainfall was considered an important disturbance for these communities, influencing rock pools structure, favoring the biomass of the chlorophyll species, typical of the shallow, turbid, and nutrient-rich ecosystems. These species that we found in rock pools are very resistant to prolonged drought. And in addition,
Ela?
É.
Oh.
Ela é ruiva?
I already muted him.
please mute your microphone. Anderson?
I think he doesn't have audio. I tried muting him, but yeah. Yeah, I got it, yeah.
Great. In addition, the anthropogenic disturbance, water abstraction, recreation use in the urban rock pools was indicated as a main impact in the urban ecosystems. We can see the high phosphorus concentrations and high phytoplankton biomass. So the difference in the conservation degree emphasized the relevance of strategies adopted in the rural zones. So where these ecosystems tend to be more distant to the human presence, the distance of the human presence is very good for the conservation, always. So the human presence causes a very big impact in the water quality, in the health of these ecosystems. And in dry zones, the water abstraction is it's essential to maintenance of the populations. So we have multiple use, and for another hand, we need to conserve this aquatic ecosystem. So it's a conflicting interests, but it's a challenge. It's the life, it's the challenge. So we can discuss about our works. And I have a question for Luisa, and Clarice can answer also. And this question was left by Maria Caldeiro Pascoal. Luisa, it's a comment of the Maria. Congrats, Luisa, your presentation looks great and take home message quite similar to the one we tried to explain in our mesocosm experiments. You already say in your conclusions that would be cool to use parafac analysis. If you have I think that step would be a key to add more information on your history. Later last year, I attended a course. It may be of your interest. She left some details about the course and about her presentation in the special session 7. You can comment? Can you comment?
Yes, I saw the comments from Maria, and I also saw her presentation in the other session. And like I said, this work that I presented here in the YouTube video was the beginning of something bigger, I can say. And this beginning was only to see the differences and to see how this difference works from natural and commercial humic substances. But after that, I like the subject, I can say. I was more interested in know and study about it. And me and Clarice, I think more Clarice than I, she told me about trophic analyze because she had already know about it from her, I think, doctoral research, I think. And we started looking for and to research about it. And we want to, in the next works, to add the perfect analyze in the results, because this analyze can return to us some quality characterization about the dissolved organic matter in general, but also dissolved organic carbon. So we want to add the perfect analyze like Maria suggests, and we are carrying this comment and carry the perfect with us, and we hope that when we could put all the ideas we are developing in this pandemic scenario, we could use perfect as a tool for make the research more complex and more detail, and give more detail about how these dissolved organic matter are and how it's this quality and everything related to this. But I think Clarice could add more information about trophic than me.
Yeah, so thank you, Luisa. I cannot add any more information because perfect is very hard. But yeah, but we appreciate the comment that Maria left us, because especially because she sent a link that's really going to help us out for the next things we have to do in the project. But and I want to congratulate also Maria for her work in the Mesocosmos study, because it was really nice, because she actually used natural and artificial humic substances in the mesocosms to see whether the natural one, which is more it's more edible and easier to crack than the human feed, which is refractory, and she compared that and saw if zooplankton was affected, and it was really interesting. And it is a little bit what we when we try to do the characterization, what the next step would be. And the most interesting thing is to try and see her results and see what Luisa is gathering now for data, and see whether we can do it as expecting changes in an hydrologic regime in these temporary ecosystems. So this is going to be nice to see, especially with Parafac, because if we can do those 3D mapping, we can actually see when the dissolved organic matter and humic substances are starting to differ when the place is filling up or drying out. So I think it's going to be really nice to add this information. Yeah, so it's going to be nice. And the scenes from the next chapters, right? So the previously and then forward. So it was nice.
very curious about the results of the mesocosmos experiments, because she used the disturbance pulse and the experiment. Very interesting. So I'm thinking about the temporary ecosystems and the complete in the period when this ecosystems is completely dry. How the effects of this dry in the cycles of the carbon or organic matter, and mainly stream droughts, because the droughts are long and more
They crack everything.
in the climate change scenarios. So
yeah, it's actually, we don't know, and it's going to be interesting when we find out, because when we talk about, because mostly DOC is coming from the soil and for us, right? Because there is this accumulation on the complete drought phase. So we have no water, a lot of organic matter cycling through the soil, and then afterwards, when it fills, it actually comes also from leaves and littoral zones when they are flooded. So we have different types of DOC entering the systems. Some of them are a little bit more decomposed and more labile, and others are more refractory. And they are all joining this pool of particulated organic carbon and then dissolved organic carbon. And they're going to be different from one ecosystem to the other, depending on the inputs we have from different sources. So it's quite nice to try and understand how this different sources of allochthonous organic carbon and different phases of this organic carbon or organic matter is going to be reactive or less reactive in different phases of the temporary ecosystem filling or drying out. So still, we don't actually know what's going on, because the sediment it's very important, though it's it's difficult to see the changes, the exchanges we have with the water column. And this is rather a big question for us. If it's going to induce error, for instance, that we were talking about this, if it's actually error that it's going to be inducing, because there's a lot of decomposition. The moment the sediment fills up with water a little bit, so we have a humid sediment, this is starting processes beyond our imagination when we're talking about carbon, right? So we have a decomposition going on. It's crazy because you have a very deepened biota over there. So doing enormous decomposition processes. So the sediment it's another black box. So
another black box.
Another black box. So, but it's interesting to see if those different sources and those different processes may actually affect higher trophic levels, like zooplankton and consumers, right? Because we expect that for phytoplankton is going to be a big problem when you have too little water and a lot of sediment and no water column. And this little water here, it has a lot of sediments on it. So there's not enough light getting in, so you don't have an autochthonous community very strong and enough to like fill up the place. But as soon as you get a little bit more water column, you have more phytoplankton, less turbidity, and a lot more of primary producers processes over there. Yeah. So we expect it to change a lot during the filling and drying out moment. And this thing between also, since this is just the thing with the sediment, because it's also giving up nutrients. And so nitrogen and phosphorus that are so important for phytoplankton are coming from this sediment, and it takes some time for it to start processing it and exchanging it with the water column. So that's why it's so important, the temporary systems. And these differences and the drying a lot, or drying less or more. Anyway.
Yeah, because
Sorry.
yeah, no, incredible answer. Because sometimes the most part of the temporary ecosystems, lengthy temporary ecosystems, are small, and the trophic web is relatively short. So the carbon is a very interesting source to zooplankton communities, for example, and can influence the composition of this community probably. So we need to expand this question for another regions.
Yeah, we need to, and we need to see. Yeah, and we need to see. Actually, we have a lot of information on temperate, and not so much on temporary ecosystems. But when we get to here in the tropics, we have a lot of temporary ecosystems that are very diverse. And Luciana, your talk showed me that, because they're even more diverse than I thought. and the cycles are entirely different from temperate regions, right? Because we don't know what's going to happen, if it's going to rain more or less, or and now with the climate change scenarios for tropical region, we actually don't know. But it's important. I think we need to understand, because the amount of endemism we have in the systems, and the amount of different ecological interactions in the systems, they're very important. And carbon, I'm sorry, is the second most important. Humification is the second most important process in the planet. So
I agree. I fully agree.
So it's just that important. So it's interesting. It's interesting because in this panel here, in this special session, we have me and Luisa talking about carbon, and you and Rafael talking about primary producers. So it was really nice.
Sign.
Yeah, it's a sign. Yeah, it's really nice.
Yeah, and in rock pools and other small ecosystems, I see very high concentrations of human substances, very high concentrations of carbon, humic substance, and but the types of the carbon forms, it's so important to study more, mainly in dry sediments. How these types persist in the dry sediments, for example.
How much there is, because even in the amount of carbon, we are not sure how much carbon, or whether carbon is incorporated or not, or actually lost during the really dry phase of the soil. So this is actually very important, because depending on the soil type, you have more or less humic substances, for instance, or organic matter, depending on the leaf as well. If there is a leaf over there, has a lot of breakage already in the soil, and it's there for ages since it fills up again. So it's really something else we have to watch. A lot of things.
We need to connect our aquatic systems with the landscape around. We need to expand this perspective to the landscape.
Yeah.
So many years we study only the aquatic systems and forgot around. So we need to expand. It's very important to contribution of the surround.
Yeah, definitely. And it's nice to see that temporary ecosystems are so reliable. They rely so much on outside subsidized. So that's the beauty about temporary ecosystems. And what makes us like, oh my God, let's study that, because there's so much exchange going on and so many things going on inside and outside, and in the pelagic zone and in the littoral zone, depending on the size of your temporary system. And it actually, in the landscape level, it changes entirely from literal, or everything's literal in one period, like for species diversity, for instance. Have everything is literal at one time period, and then it rains a lot, then you have a pelagic region and a littoral region. And then this is amazing. Like this variation, it permits a lot of questions and interesting ecological assumptions, and it's very nice. But anyway, I've stopped talking now. Luisa even gave up.
Big challenge.
Go ahead, Rafa.
Big challenge. Rafa?
No, I also have a concern about that trade-off that you just established here between photosynthesis and productivity and dissolved organic carbon, because from the perspective of climate change, we will improve the irradiation, solar radiation and light irradiation in many parts of the world, but also photosynthesis is also influenced by light radiation. So both the productivity, the production of dissolved organic carbon will be increased, maybe, and also the degradation by photooxidation. So what should we expect from this balance?
Yeah, Rafa, what we are seeing, we have a lot of studies on climate change and DOC increase in temperate regions, which is a very important and expected relation, but regarding temperature increase. Here in the tropics, since temperatures not can have this gap as in the temperate zones, the problem is rainfall and light radiation in the ecosystem. But the thing is, what we can expect is that we have photodegradation, an increase in photodegradation of DOC in aquatic ecosystems if UV increases. But this UV change and photodegradation of the carbon can actually be good for phytoplankton, since it may transform to very good labile carbon for phytoplankton, like affecting priming effects in this community. And actually, we have also, with the increase of UVA and UVB and the increase of carbon in the systems, some articles already saying that we might have like a sunscreen process for phytoplankton. So the UVA increase in temperate zones might actually be a problem for phytoplankton, and the photodegradation of carbon and the more labile carbon may actually be like a sunscreen factor, helping the photosynthesis apparatus not to be broken for a lot of UVA incidents. So there's a lot of stochastic events, and this can also be interesting when we try to see if there is a lot of chances for extreme events on rainfall, for instance. Then you have actually more carbon coming in the system. So if we isolate this part of that, like rainfall, let's talk about rainfall. So rainfall is decreasing DOC incident runoff to the aquatic system. And this is actually going to be bad for phytoplankton, because then it's going to increase DOC and more humic substance over there. So there'll probably be a little bit of depletion on primary productivity. So there's a lot of, if we try to separate the boxes, then okay, so this can cause a problem for phytoplankton, but actually this cannot actually be such a problem. So we have to try, and this is for future studies, maybe trying to gather in maybe in experimental studies and try to block them up together, you know, see whether, let's increase the input of DOC over here and actually put a lot of light in here as well. So if there is a trade-off between the two factors that we might be expecting for climate change here in tropical areas. So, yeah, I think that's it.
It's very interesting to explore these trade-offs a little more deeply.
Rafa, I actually have a question for you. Okay. Sorry, Lu. I have a question for you now regarding, I have a question, I didn't put it there because I wanted to ask him, but let me see. Because you said it was really important in your paper, and it was really nice to see the semi-arid ponds and again, more diverse landscapes that we are actually accustomed here in the Southeast. But it wasn't entirely clear for me, actually, in your work. I know it's preliminary work. The reason why you decided to measure in two different time frames. Actually, it became clear, but maybe you can clarify it here as well, because you measured at 3 p.m. at 3 p.m., right, and at 5 a.m. in the morning, and you noticed some differences, right? Can you comment on that?
It's just a question of how much the ponds will be retain temperature and retain light absorption. Like so, in the beginning of the morning, the phytoplankton was exposed to no light, like total dark, and then in the afternoon exposed to the peak of solar irradiation. So I want to explore these two scenarios. The influence of light in chlorophyll. Because this is experimentally tested for some species in temperate regions, but it's not much addressed in tropical systems with high solar variation. I think temperature is maybe not the main factor driving this, because it's hot all the time it's warm all the time in Brazil and tropical systems, but maybe light is important.
Definitely. Rafa, I think Luisa also has a question for you.
Okay.
Hi, first of all, congrats for your work. I really like it. And the stratification that you present was awesome, because when I studied about this kind of stratification, was always related to marine environments, not in aquatic ecosystems. So it was really, really awesome. But the question that I had, that I want to ask to you, is that you highlight the climate change effect in your system. And Luciana also did that, but I'm going to focus on your work. And I want to know if you want to add the predictions from IPCC reports for semi-arid regions, because like Clarice said, the climate change predictions are very heterogeneous from world. We have a lot of predictions from temperate region, but here in tropic there are some difference. So I want to know if you want to add this kind of predictions, mainly rainfall pattern and temperature, using a kind of time tracking, I think, because we are working with temporal ecosystems. Wetland is more me and Clarice. So I want to know if you want to add this kind of approach in your work. Luciana can also ask and answer this, because I think this can be put in this work, in her work too. Sorry, but I want to know what is your opinion about this.
Thank you, Luisa. For further investigation, I would do some experiments in laboratory with radiation metrics. I would like to measure the intensity of light radiation to this in experimental approach. But these changes in these large changes regarding climate change would be not addressed in this, maybe not for now, because we have some things to accomplish in PhD, so it's too much things to do so. But we can think about that, maybe collaborate with you and Clarice and Luciana and Mel's one too. It's interesting question, but it's very, I think it's a very important and difficult to investigate alone. So we need to collaborate in that.
Very great. It's interesting because in brief, we have more questions than answers in regard to the temporary ecosystems. It's a big challenge to work in these ecosystems and extremely vulnerable to the climate change. And on the other hand, extremely vulnerable to human impacts and it's so hard, yes, to separate the effects of climate change and human impacts in short scales. So we need to adopt different ways to understand better the functioning of these ecosystems in the landscape. And in my view, the landscape is the big challenge to understand the landscape and connections of the temporary ecosystems between them and with the landscape, other compartments. No, it's a very big challenge. So we have some questions. Clarice, what do you think?
No, the question?
I don't know. We can conduct to speak about the perspectives and about our next Congress, for example, before to finish our session. Yeah. Yes. No. Luisa. Oh, Luisa have another question or not?
No,
I don't think so.
I already did it to Rafael.
Okay.
Yeah. So
I don't know, but in my viewpoint, this is the first step to discussion. We introduce the issue and some issues, important issues, and we need to deep in these concepts, in the experimental designers and perspectives of the studies of the carbon and other substance in the temporary ecosystems, including nutrients and energy flow. So I'd like to invite you to participate to the next CUA Congress. Will be held in August, fully online. And in there, I, Clarice, and Egor Zaderev will be held a special session about temporary ecosystems. So Luisa, Rafael, and other people present, you invited to participate. And after that, will be held a meeting, working group meeting, Limnology of Drylands. And so I will invite Clarice and your team to discuss specifically about the human substance and carbon in temporary ecosystems. Despite the dryland zones, we can expand our viewpoint in other directions, mainly in temporary ecosystems in tropical regions, and including in humid regions. May be a very good, important ecosystem to understand the climate change scenarios and response to the climate change or global warming. So what do you think, Clarice?
Yeah, I think it's great. It's going to be a nice, also very nice special session. And we have to discuss that. We have to discuss so many things because what I'm seeing is a difficulty because for instance, we're working with also in my postdoc, we're working with, I think I already told you that, Luciana. We're working with temporary systems in the Amazon also. And they're a little bit similar to rock pools, but not as much because they are in a very mineral basin, but they are ponds that fill up to like 40 centimeters depth. And what the problem is, we have a lot of information, but the data is very sparse. And the research started like 15 years ago, and we started doing this kind of sampling, and now we're doing some other kind of sampling. So this is another thing we should discuss here and over there in SIL, is that how difficult it is to unite data on those temporary systems. Because it's funny because it's so the research we are conducting is so old-fashioned, but still for temporary systems they're so needed. And like, okay, we need to monitor annually those systems. I need to see it dry out. I need to see when it dries out, or what happens when it's dry. And actually, we didn't have this interest before because, okay, we get to a place and there's no lake, there's no pond, there's nothing. So there's no lake, so let's go. We never try to investigate what's going to happen as soon as there is a little pond, like a one meter squared area pond. What's going on there? So this is hard because there's a lot of gaps on the data. And I don't know whether you're seeing that with your meta-analysis. You mentioned once that you're trying to do the meta-analysis. And if it was a problem for you, and we should discuss that, not only data gap, but data quality as well for communities, because not a lot of people are sampling those very diverse communities. So this is something else we should gather in the working group here with everyone in the conference as well. So, yeah.
That's a great opportunity. We start some studies with symptomatic and meta-analysis approach, mainly to try to fill gaps, to filling gaps. So, but it's a very big challenge. But we can see some trends in the global scale. So I like to think about the symptomatic studies and meta-analysis to understand the distribution of temporary ecosystems and main gaps in the studies performed in these specific ecosystems. So we can try to together to identify some aspects. I can send for you and everyone to our ideas, share our ideas, because it's very important to increase the understand of the distribution and flow of energy and nutrients, effects of stream droughts in the process and the communities, because our focus the most part of the time is in communities, so the process need more attention. It's an opportunity to deep in this aspect, very important aspect. Yeah.
Yeah, it's very important. Thank you very much, Luciana. It's been a pleasure working with you in this sessions, and I think it's going to be a nice collaboration with the young Rafael and Luisa and everyone here in this room.
Very big opportunity. Thank you, Erica. Thank you, Luisa, Rafael, and everyone to watching, to discuss. And I'm very happy with our session. And I wait for the next meetings. Clarice, thank you so much.
It was really nice. Thank you.
I learned a lot today. Thank you.
Me too. Me too.